Hello everyone, my name is Professor Shamsul Hayat from Department of Botany, Aligarh Muslim University, Aligarh. I, along with my colleague Professor Qazi Fariduddin, welcome you all for this course of plant physiology and metabolism. For this course, I was assisted by Ms. Yamshi Arif and Bisma Hilal, a PhD scholar in my laboratory. Coming to the introduction of plant hormone or regulators, these are chemical messengers that are produced in one cell and modulate cellular processes in another cell by interacting with specific proteins that functions as receptors linked to cellular transduction pathway. These are produced in small quantities by a plant and then transported elsewhere for use. Hormone is derived from the Greek word hormene, which means to excite. The plant development is regulated by the 6 major classes of plant hormone. These are auxins, gibberellins, cytokinin, ethylene, abscisic acid and breast-steroids. However, there are some signaling molecules that as a hormone like jasmonic acid and salicylic acid that play roles in resistance to pathogen and defense against herbivores. Moreover, strigolactone regulates the outgrowth of lateral buds. Now we will discuss about first discovered hormone, that is auxin. It is derived from the Greek word auxin, that means to grow or increase. Auxins may be defined as growth promoting substances which promote growth along the vertical axis when applied in low concentration to the shoot of the plant. Auxins play an important role in plant growth and developmental processes as induces stem elongation, apical dominance, root initiation, fruit development as well as the meristem development. So, if you look, when conducted an experiment where they meticulously removed the tips of oat coleoptides, the protective sheath covering the emerging shoot, astonishingly the bending response was affected, suggesting that something in the tip influenced the plant growth. To test this hypothesis, he took extracted substances from the tips and placed them on the cut surfaces of the coleoptides. Remarkably, the bending response was promptly restored, thus successfully isolated this mysterious substance from the tip and named it as auxin. Moving ahead, there are several naturally auxin such as indole acid, indole 3-butyric acid, phenylacetic acid, 4-chlorindole acid. However, there are several synthetic auxins such as indole 3-propionic acid, alpha-naphthalene acetic acid, 2,4-dichlorophenoxyacetic acid and 2-methoxy 3,6-dichlorobenzoic acid, also called as dicamba, which is a herbicide. These belongs to the synthetic auxin. So, we can look in a schematic diagram of nonpolar and polar auxin transport in Arabidopsis thaliana. Auxin is redistributed throughout all aerial and underground plant body through first, that is nonpolar transport in phloem, which is marked by the blue arrows, and slow, that is cell to cell polar auxin transport, also which is indicated by the green and black arrows. The polar auxin transport is expressed as a preferential movement of auxin from top to bottom in plant axis, that is in basipetal manner. Moreover, root also exhibit basipetal transport from root tip to the base. Polar transport of auxin is highly temperature sensitive. There are several auxin transport inhibitors such as cyanide, 2,4-dinitrophenol, 2,3,5-tindazole benzoic acid. Moving forward, this figure shows the presumptive pathway for the IA biosynthesis in plants. Green arrows indicate the tryptophan synthetic pathway in chloroplast. A thin dashed black arrows denotes the tryptophan independent pathway. Blue arrows indicate step for which the gene and enzymatic functions are known in the tryptophan dependent IA biosynthetic pathway. Mustard colored arrows indicate the Brassicaceae species specific pathway. IA is synthesized basically from a amino acid that is known as a tryptophan. Tryptophan is converted enzymatically into indole acetaldehyde by two different routes, first by indole 3 pyruvic acid and second by tryptamine. However, in the member of Brassicaceae family, tryptophan is converted into indole acetol dioxime into indole acetonitrile directly or through glucoside formation. Then indole acetonitrile is finally converted into IA. So, auxin basically promote growth in plant by cell elongation mechanism. As you look, auxin includes two types of response in plant, that is, one is the fast response and another is the slow response. Fast response appear basically within few seconds or minutes, as well as the slow response appear within minutes or hours. Cell elongation is basically comprises of two events, that is, first one is the loosening of the cell wall and the second one is the maintenance of the secondary structure of the cell wall. As illustrated in this figure, that cell wall loosening involves breaking cell enlarged arrangement of bonds in cell wall polysaccharides. The convincing explanation is offered by acid growth hypothesis, which suggests that in increase in acidity near to the cell wall is caused by basically this hydrogen ATPase. So, in the plasma membrane that pump proton into the cell wall. Therefore, the accumulation of hydrogen ion lowers the pH, which activates enzyme that facilitate the breaking of acid labile bond such as hydrogen bonds in cell wall polysaccharide and ultimately leading to the cell wall loosening. Here, as shown in the figure, that the maintenance of the secondary structure of the cell wall, or you can say the maintenance of the cell loosening, is induced by the auxin, which involves an enhancement in the uptake and accumulation of solutes, thereby elevating the osmotic potential within the cell cytoplasm. Therefore, the increase in osmotic potential, which results in turgidity, aligning with the elongation of the cell. Moving ahead to physiological roles of auxin, auxin basically improves several physiological responses such as cell elongation and division, dormancy, control of premature fruit drop and delay in the leaf abscission, apical dominance, parthenocarpy, adventitious root formation, sex expression, shortening of internodes, and prevents the lodging. So, as you can look, this slide basically displays the regulatory role of auxin. Auxin increases the cell division. Formation of chylus is the best example is shown. Chylus growth is dependent on auxin level. High auxin ratio induces root formation, while low level induces shoot formation. Moving to the next role, that is the cell elongation, which was induced by auxin. it is also directly proportional to the auxin concentration. Moreover, low concentration promotes the root elongation, however, high concentration reduces it. Auxin also stimulates the growth of the adventitious roots. Moving ahead, the next role that auxin maintains the apical dominance, such as the tea plant where lateral bud growth is suppressed by the apical buds. Auxin delay the senescence in leaves and also promote the shortening of the internode. For example, apple and pear have two types of branches, that is dwarf and long. Only dwarf shoot bears fruit, and the spraying of Na on stem apices favors the dwarf shoot development. As you can look, auxin also prolong the dormancy. It was observed that application of naphthalene acetic acid retard the sprouting of the tomato. Then next one is the parthenocarpy. It is a development of fruit without the fertilization. However, auxin induces parthenocarpy in apples, tomatoes, banana, etc. Lower internodes in stem of oat, paddy, wheat are long and weak. As a result, the plant bend down. It is called basically the lodging. However, auxin reduces lodging as induced cell division, which leads to the formation of xylem that give basically the mechanical support to plant. In cucurbits, male flowers are more than female. However, application of auxin reduces the number of male flowers and increases the number of female flowers. Auxin application reduces the premature fruit drop and also used to eradicate the weeds. 2,4-D and 2,4,5-T are used as a weed killer. Therefore, auxin can play an important role in sustainable agriculture. The auxin was also used as a weapon of biological war in the US-Vietnam War. As it is clearly displayed that ultimate goal of any research on the molecular mechanism of hormone action is basically the signaling transduction pathway. So, from receptor binding to physiological response. However, auxin may interact with auxin or the calcium ions, which in turn calmodulin, a protein which regulates many processes in plants, animal and microbes. Basically, the hormone binds with the receptor, and after binding there is a various functions, or the you can say the reaction are produced and the second messengers can be there, and then finally there is a some activation of the cellular response is occurred. The primary the primary receptors for auxin are recognized as complexes consisting of a soluble protein from the TIR1-AFB family and a number of the larger AUXIA family of transcriptional repressor protein. TIR1-AFB proteins are named after the first identified number of the plants known as transport inhibitor resistant 1. These proteins are broadly classified as auxin F-box binding proteins and serve as F-box protein within an SCF ubiquitin E3 ligase complex facilitating ATP dependent catalysis. In the auxin signaling pathway, basically the TIR or AFB genes in the auxin receptor gene family which encode for F-box components of the SCF complex. This complex play a role in degrading auxin indole 3-acetic acid, that is auxin/IA repressor, regulating auxin response gene transcription as shown in the figure. Auxin responsive genes typically features auxin response element that is called as AUX binding site in their promoter region and transcriptor factor known as a auxin response factor. These bind to the RF motif to either stimulate or repress transcription. Therefore, on activating transcription, ARF form homodimers through conserved interaction domain and recruit chromatin remodeling factors. In low auxin concentration, auxin IA repressor protein with interaction domain similar to ARF form heterodimers with ARF leading to the repression of the transcriptional activation. So, this is all for this class. In next class, we will discuss about the cytokinin. Thank you.